It's also going to utilize the enzyme RNA polymerase. Now, if DNA polymerase made strands of DNA nucleotides, then you could assume that RNA polymerase would make strands of RNA nucleotides. The one difference between DNA polymerase and RNA polymerase is that RNA polymerase is going to use the uracil nucleotide instead of thymine. And you could agree that we're probably going to be obviously reading the DNA to make this possible. So we could state that transcription will start at a promoter region that's on the DNA and will stop at the terminator region. So what I want to go over on this little drawing that's right here is, of course, you see your DNA strand, these two original pieces. Agreed? So what we have here is we have the binding of this enzyme, which, of course, this enzyme is going to be RNA polymerase. Agreed? And you'll see in this picture below that we've started to make this strand of RNA. Agreed? So this actually would be what I would call mRNA. Possibly, though, it might be pre-mRNA. It's actually, if it's a eukaryotic cell, it's a guarantee it's probably going to be pre-mRNA. It still needs to be modified. Now, you can still, if you remember from chapter 16, you can only write nucleotides in which direction? 3 to 5 or 5 to 3?
5 to 3.
So, still 5 to 3, just as it's been. That means that this has to be a 5' end and this has to be a 3' end. Okay? And that would mean that the original DNA strand, if we think about it, has to be 5' there and 3' there. That would be a 5' there and a 3' there. Right? You'll notice that the RNA polymerase has started in a site called the promoter site. So it's start point. It's going to finish the sequence at the terminator site. And I want to make sure we still see the 5 to 3 designation of the writing. Bless you. And though you are not required to draw it, I want you to see that at the end, you've got your 2 DNA strands, still as normal as before, right? And now you have your little strand of mRNA. It's really the same thing as going to the library, using the encyclopedia, and then having your notes afterwards. When you write notes, you jot down some ideas, maybe on a scratch sheet of paper on little note cards from an encyclopedia or whatever other resources you're using. The resource isn't torn up. It's still usable again. Your DNA is that resource. You'll notice that the polymerase is off and reusable as well. In fact, if you think about it, there's really nothing stopping it from copying, or transcribing, I should say, better put, the same segment of DNA again. Now, most likely, and this I want to make sure we get, so listen closely, most likely, this sequence of mRNA is going to code for one single gene, or multiple related genes. So I want to make sure we get that this mRNA sequence is representing one gene or several genes that are all very related. It's like buying it's either the one gene is going to operate by itself, or maybe sometimes you need a package of genes, a package deal, to do this specific sequence. But regardless, this has potential to become, or to be read into, translated into a polypeptide down the road somewhere. Good? Okay. We 100% on that? Okay, awesome. We could view this overall process of transcription as being about three major steps or three major events. The first would be to begin it or initiate it, then to elongate the RNA strand, and then finally to terminate or end the production of the mRNA strand. So let's talk about the first sequence, the first step. Initiation. So to begin the process of making RNA, probably an mRNA strand. Transcription factors. Will help the binding of RNA polymerase onto the DNA. The combination of all of the transcription factors, and there's multiple of them. Combination of the transcription factors and RNA polymerase is called the transcription initiation complex. There's a box or an area that's made of repeating units of thymine and adenine. TA, 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 repeating unit is important. In forming this complex. Bless you. All of these events occur at the promoter. Question. Bases are T, A, G, A, T, G, and C on DNA. So repeating units of T and then A and then T and then A is a sign of that's where we're going to start this, where the promoter region starts basically.
So it's the opposite of like the EG or like the CG.
I'm sorry. Do it.
Like would the opposite, if it was like CG or UG, would be like the ending side?
No, no. So I'm gonna get you to copy down another picture because I know you like them. Just want one though. This one will take not too long to do. It's this little picture right here.